The nervous system allows a person to detect stimuli. Draw one line from each stimulus to the sense organ that detects the stimulus, and this question is worth 2 marks. First of all, make sure you understand what is meant by stimulus. This is a change in the environment. So what you have to do is match the boxes on the left to 2 of the boxes on the right. So the chemicals are detected by your tongue, it's referring to what you're tasting, so you draw a line between those 2, and the second box is light, you detect that with your eye. Moving a hand away from a hot object is an example of a reflex action. What is a reflex action? And this question is worth 2 marks. Well, a reflex reaction is an unconscious response to danger, so you can describe it as a rapid, involuntary response to protect the body from harm. A muscle in the arm moves the hand away from a hot object. How does the arm muscle do this? Tick 1 box, and this question is worth 1 mark. The correct answer is that the muscle contracts, so you tick the first box. Two students investigated the effect of drinking coffee on reaction time. This is the method used. Student A holds a meter rule just above student B's hand, as shown in figure 1. Student A lets go of the meter rule. Student B catches the meter rule as quickly as possible. Student A writes down the reading from the scale on the meter rule. Student A and B repeat steps 1 to 4 another 4 times. Student B then drinks a cup of coffee, and after 15 minutes, students A and B repeat the steps 1 to 5. So this is figure 1 showing how this ruler is dropped. So student A will hold the ruler above student B's hand and then will let go, and student B has to try and catch it as quickly as possible. And from looking at the image on the right, you can see what qualifies as a reading. So they've said that it is 18 centimetres that student B has caught it at, and 18 is the number that is just about covered by their thumb. So that's how you want to read it in the future. Table 1 shows some of the results. So here we've got table 1, and it's showing us the reading from the scale on the meter rule in centimetres, and we've got before drinking coffee on the left and after drinking coffee on the right, and then the very left hand column shows which test it is, so that just shows us that they've done repeats. Figure 2 shows the results after drinking the coffee for tests 3, 4 and 5. So here's figure 2, we've got the images of the hand holding the ruler for each of these tests. And we have to use these images to complete table 1. Where it says use the results from figure 2, it wants us to read that number that the thumb is just about covering. And this question is worth 2 marks. So let's first look at the numbers we can see. For test number 3, we can see the number 9, and the numbers are increasing as you go up the ruler. So the number just below the number 9 where the thumb is will be number 8. So we record in the table 8 centimetres. For test 4, we can clearly see the number 12, which means the number below that the thumb is just covering is the number 11. So that's the next one we can record. And finally, test 5, we can see the number 14, which means the number just covered by the thumb is number 13. So that's the final thing to enter into the table. The students made the following conclusion. Drinking coffee speeds up reactions. Give evidence from table 1 to support the student's conclusion, and this question is worth 1 mark. So what you need to do is compare the distances that the ruler has fallen in table 1, and if it's fallen further, we know the reaction time is longer. By looking at table 1, you can quickly see that all of the numbers on the left are greater than those on the right. So 18 is greater than 10, 21 is greater than 14, 15 is greater than 8, 12 is greater than 11, and 19 is greater than 13. And you get that mark for saying that after drinking coffee, the ruler does not fall as far. The student's conclusion may not be valid. Suggest 2 improvements the students could make to their method. and this question is worth 2 marks, so 1 mark per improvement. Well, they can carry out more repeats, so this gives them a better average. They can test more students, this gives a greater spread of data that applies to more people. They can use a ruler with a more precise scale, so it's got smaller divisions. They can also make sure that they are always dropping the ruler from the same distance above student B's hand, and also make sure that student B remains stationary, that they're not moving when student A might be about to drop the ruler. The shape of a person's earlobes is controlled by a gene. Figure 3 shows 2 types of earlobe. So here we have figure 3 just below. We've got the free earlobe on the left, and you can see that it's not attached to the neck. And then on the right you've got the attached earlobe, and you can see there isn't a gap between the bottom of the ear and the person's neck. A dominant allele codes for free earlobes. What is a dominant allele? Tick 1 box, and this question is worth 1 mark. So the rule with a dominant allele is that it is always expressed even if there is only one of them. So what you've got to do is look for the one of the statements that aligns with this. The first statement says an allele expressed even if a person only has 1 copy of the allele, which aligns with our definition. The second one says an allele expressed only if a person has 2 copies of the allele, so this is a recessive one, so that's not correct. The third says an allele expressed only if a person has no recessive allele, that's false. And the last one says an allele expressed only if it is inherited from the male parent, that's not correct either. So the box you have to tick is the first one, an allele expressed even if a person only has 1 copy of the allele. A man with 3 earlobes and a woman with attached earlobes have children together. Complete figure 4 to show the possible genotypes of the children. Use the symbols capital E for the allele for free earlobes and lowercase e for the allele for attached earlobes. And this question is worth 2 marks. So what you have to do is fill in the letters that correspond with the allele of the man and the allele of the woman. So the top right box we get a capital E from the man and a lowercase e from the woman. And then the lower 2 boxes we get a small e from the woman and a small e from the man in both cases. What is the probability that one of the children would have attached earlobes? Use figure 4 and tick 1 box, and this question is worth 1 mark. So we have a copy of figure 4 below to help us out, and we remember that the allele for attached earlobes is the lowercase e, and it means that we need 2 of them because it is recessive. So we look at the diagram and we see which boxes have 2 lowercase e's in them, and we can see it is the 2 at the bottom. So 2 cases out of 4 will have that homozygous recessive allele combination, and that gives us a probability of 0.5. Figure 5 shows the inheritance of the sex chromosomes X and Y. Complete figure 5 to show the sex chromosomes in the gametes of the man and the woman, and this question is worth 2 marks. So figure 5 is just below, and we have to fill in the sex chromosomes for the man and the woman. A woman has the chromosome combination XX, and a man has the chromosome combination XY. Calculate the probability that the man and the woman's next child will be a girl with attached earlobes. Use the equation probability of a girl with attached earlobes equals probability of attached earlobes multiplied by the probability of being a girl. And this question is worth 2 marks. So we have our 2 genetic cross diagrams here on the left, and we can start by figuring out the probability of the child being a girl. So you can see for the woman the chromosomes have to be XX. So we've got to look in the diagram and see how many of them are X and X, and that's the top 2. So that's 2 out of 4, and that gives us a probability of 0.5. Then we repeat this process to find the probability of having attached earlobes. And remember that allele is the lowercase e, it's recessive, and we need 2 of them together, which is the bottom 2 in the other genetic cross. Again, we get 2 out of 4 equal to 0.5, and we use our formula to multiply the two together. So 0.5 multiplied by 0.5 gives us 0.25, and that's your final answer. The mark scheme may also allow you to give this as a percentage, so 25%, or as a fraction, 1/4. Figure 6 shows the production of sperm cells in humans. So the diagram is figure 6. We start with cell A, and then it shows the cell grows and copies genetic material, and then it divides and divides again to produce sperm cells. Cell A is a normal body cell. How many chromosomes are there in cell A? Tick one box, and this question is worth 1 mark. So the options we're given are 23, 46, 48 and 92. The correct answer is 46, and the number 23 is worth remembering as the number of pairs of chromosomes. What is the mass of DNA in cell E? Tick one box, and this question is worth 1 mark. So figure 6 is really useful for working this out. The first thing you want to do is identify cell E, which is the bottom left, and then we can identify cell A because that's what all of our answers are in reference to. So if we think of the mass of DNA in cell A as X, we can see that cell B will be 2X in terms of the mass of DNA because the genetic material has all been copied. Then when cell B divides into 2, that value splits again, so we're back to X DNA in C and D. And then cells C and D divide, so that X gets split in half. So E will have X over 2 in terms of the mass of DNA. And that means it will have half of the mass of DNA in cell A. So the second box down is the one you want to tick. What type of cell division produces sperm cells? Tick one box, and this question is worth 1 mark. So if we look at the options we're given, binary fission we know is not correct because that's how bacteria divide. Differentiation is the process by which a stem cell becomes specialised, so by process of elimination, meiosis can be the only answer. Sometimes there are errors in copying the genetic material. What term describes an error in the genetic material? Tick one box, and this question is worth 1 mark. So looking at these terms, we know it's not absorption because that is a completely different process. Fertilisation is the process by which the sperm and the egg nuclei fuse. Mitosis is a form of cell division, so the only one that could be is mutation, which is a change in genetic material. A woman has 3 children, aged 4, 6 and 9 years. Why are the children not genetically identical? And this question is worth 2 marks. So this is because gametes are all different, and for each of these children, a different egg or sperm was used each time, and each gamete has different genetic information or different alleles and DNA. You can also get a mark for saying that the genes come from 2 different people. There are 2 parents, and there isn't just one single parent cell. In sexual reproduction, a sperm cell fuses with an egg cell to form a new single cell. An embryo develops from this single cell. The cell divides 3 times to produce an embryo. How many cells are there in the embryo after 3 cell divisions? Tick one box, and this question is worth 1 mark. So the information you need here is that the cell has divided 3 times, and you can figure out how many daughter cells there will be by drawing a diagram, and it represents 2 cubed. So after the first division, there are 2 daughter cells. Then in the second division, both of those daughter cells divide, so you have 4, and then those 4 divide again in the third division, and you get 8 overall. So the box you tick is the number 8. Figure 7 shows a different human embryo. So the image is figure 7, and we can see that we have this label X, which is showing the length of the embryo. And it says, measure the image length X on figure 7. Give your answer in millimetres, and this question is worth 1 mark. So the image will have been printed a specific size on your exam paper, and when you measure it, you get an answer of 40 millimetres. The image in figure 7 has been magnified 500 times. Calculate the real length of the embryo. Use the equation real length of embryo equals image length over magnification, and give your answer in micrometers. So 1 millimetre equals 1,000 micrometers, that's the conversion they give you, and this question is worth 3 marks. So we figured out our actual image length in the previous question. We know that it is 40 millimetres. This question itself gives us the magnification, so we know it's been magnified 500 times. Next, we want to convert our measurement in millimetres to micrometers, so 40 millimetres times by 1,000 gives us 40,000. Finally, we apply our equation, so the image length 40,000 divided by the magnification 500 gives us a final answer of 80 micrometers. So here you would get 1 mark for your conversion to micrometers, 1 mark for using the equation, and 1 mark for that final answer. The embryo may not implant in the lining of the uterus. The embryo will then be lost from the woman's body several days later. Explain why the woman may not notice this has happened, and this question is worth 2 marks. So the reason this happens is because the embryo is very, very small, and it is not necessarily felt or seen by the woman when it is lost from her body. The mark scheme will also allow you to make a comment on how the embryo is lost in the woman's normal menstrual flow. Gardeners sometimes make compost heaps from dead plant material. The dead plants decay in the compost heap. Figure 8 shows a compost heap. So below we've got figure 8, and you can see the layers of dead plant material are the thicker bands, and the thin layers of soil are the thinner, darker bands. The thin layers of soil contains organisms that cause decay. Which 2 types of organism cause decay? Tick 2 boxes, and this question is worth 2 marks. So going down the list, we know that bacteria is correct, those cause decay, and we also know that fungi is correct. So the ones you want to tick are the top 2. The rate of decay in the compost heap depends on several environmental factors. Explain how the rate of decay would be affected by an increase in oxygen concentration or a temperature increase from 5 degrees to 25 degrees. And this question is worth 3 marks. So both of the factors that they've given us will increase the rate of decay, and this is because increasing the oxygen concentration increases the oxygen available for the decomposers to respire aerobically, and then increasing the temperature increases the rate of reactions, and this is because it increases enzyme activity. So here you would get your first mark for saying that both increase the rate, you get your second mark for talking about respiration, and the third mark for saying that increased temperature causes faster reactions. Give 1 environmental factor needed for decay. Do not refer to oxygen or temperature in your answer, and this question is worth 1 mark. The answer is water, and you can also put H2O. Dead plant material can also be decayed in a biogas generator. Figure 9 shows the percentages of the gases found in a sample of biogas. So below is figure 9, a pie chart, which shows that other gases are 5%, carbon dioxide is 35%, and gas X is the remainder. Gas X is the main fuel gas found in the biogas. What is gas X? Tick 1 box, and this question is worth 1 mark. And the answer to this is methane. That's the predominant gas in biogas. What is the percentage of gas X in the biogas? And this question is worth 1 mark. So we know the total of our pie chart is equal to 100%. We are given the values of 5% and 35%, and we know that whatever is left over must be gas X. So we do 100 minus 5% minus 35%, and that gives us an answer of 60%. The dead plant material in the compost heap and biogas generator does not decay completely. Explain why a farmer might spread the remaining dead plant material onto his fields, and this question is worth 2 marks. So the reason the farmer does this is to make the plants or the crops that they're trying to grow faster, and this is due to the dead plant material that they've spread over the fields containing lots of mineral ions which are good for the plants. And the mark scheme will allow you to say this in quite a few ways. So you can say that it's a fertiliser, you can also mention that it improves drainage and suppresses weed growth, or you could say that it is an insulator and it will protect the plants from things like frost, and it improves the soil structure. Figure 10 shows a flightless bird called the dodo, Raphus cucullatus. The dodo was 1 metre tall, had a mass of 20 kilograms, lived in rainforests on a tropical island, ate fruits, made its nests on the ground, and a female dodo laid only 1 egg each year. Humans arrived on the island in the year 1507. By 1681, the dodo was extinct. What is the genus of the dodo? Tick 1 box, and this question is worth 1 mark. Looking at the previous page, we see the name of the dodo is Raphus cucullatus, and the first term in this is the genus and the second is the species. Therefore, the answer to our question is Raphus, and that is the last option in the multiple choice, so you tick that one. Before the arrival of the humans, there were no other large animals living on the island. Suggest 2 reasons why the dodo became extinct soon after the arrival of the humans, and this question is worth 2 marks. So there are many potential reasons for this. One is that the humans hunted and killed the dodos for food, and the same can be said about the dodo's eggs as well. They may have been collected or eaten by humans. You could also say that the humans ate the dodo's food, so this is an example of competition. You could say that the animals brought by the humans ate the dodos, so these would be creatures like dogs that were brought over on human ships. You could say that diseases are introduced by humans or by the animals that the humans have bought, and the dodos would not necessarily be adapted to survive this. And lastly, you could say that the humans destroyed the habitats that the dodos were living in. Today, humans are cutting down large areas of tropical rainforests. Suggest 1 use of the land after the trees have been removed, and this question is worth 1 mark. There are many potential answers here. One is growing crops or biofuels. One is grazing animals. Another is building houses for people to live in. You could say quarrying or mining for materials, and dumping waste, so for landfills. Why does the removal of trees cause an increase in carbon dioxide in the atmosphere? Tick 2 boxes, and this question is worth 2 marks. You can work out the correct answer by working your way down the list. So the first option isn't correct because fewer animals would mean fewer exhalations of carbon dioxide, so that would decrease carbon dioxide levels. The next option, however, is correct because less photosynthesis means that less carbon dioxide is being taken in from the atmosphere, and you can see that from looking at the photosynthesis equation, which has carbon dioxide as a reactant. The next option is incorrect because we're talking about the removal of trees, and trees don't respire as much as they photosynthesize. And even if we were talking about the effects of animals that would be living among the trees, less respiration would decrease the carbon dioxide in the atmosphere, not increase it. The soil drying out is incorrect because it's less relevant, it's talking about water, not carbon dioxide. But the final option is correct because trees being burned is a combustion reaction, and a product of combustion is carbon dioxide. What effect would an increase in carbon dioxide in the atmosphere have on global air temperature? Tick one box, and this question is worth 1 mark. So when we increase carbon dioxide, we are increasing the amount of greenhouse gas in the atmosphere, and more greenhouse gases mean we get more of the greenhouse effect, which is what contributes to global warming. Therefore, the global air temperature will increase. Sustainable forestry reduces the harmful effects of cutting down trees on the environment. Figure 11 shows a method of sustainable forestry. Numbers 1 through 9 show different parts of a rainforest. So here is figure 11, it shows a map of the rainforest, and each of these numbers represents a different section. The trees are cut down in the sequence 1 to 9. The trees are cut down in only one area at a time. It takes 30 years to cut down the trees in each area. The trees in the old growth area are never cut down, so that's that section in the middle of the forest. How many years would it take to cut down the trees in all of the numbered areas in figure 11? And this question is worth 2 marks. So remember from the diagram we have 9 sections, and it tells us that each section takes 30 years to cut down. So this means that we have to multiply 9 and 30 to get our final answer, and that is 270. The rainforest contains 750 species of trees, 400 species of birds, 150 species of butterflies, and many other species of plants and animals. Explain how the pattern of cutting down trees shown in figure 11 stops the biodiversity of the rainforest being reduced. And this question is worth 4 marks. So if we look at a simple diagram of how this rainforest is split up, we can see that there are lots of different sections, and only one section is cut down at a time. This means that if that section marked at the bottom is cut down, the animals from there can simply move on to the next section, and if that one is cut down, then they can move on to the next, and so on. We also know that it takes 30 years, it takes a long time to cut down that one section, so by the time we get to that first section marked with the X again, it will have regenerated, and the animals from other sections that were being cut down will have moved back there and been able to live there until it came time for that one to go down again several generations later. We also know that that central section is never cut down, and this means that it can be regarded as a source of recolonising organisms. So now we can begin to formulate our answer. We can say that displaced animals, so that's those that come from the area that was being cut down, can move to the adjacent zones where there is still suitable habitat available because the trees there have not been cut down. You can also talk about the fact that seeds from forested areas are still being produced and they're still being blown around the rainforest by the wind, and this means that the area that has had all of the trees cut down will receive seeds from the other areas and will therefore regenerate later. And you can make a statement that there is sufficient time for the regeneration and for the new plants and trees to grow because it does take 30 years to even cut down one section. As these new plants and trees grow back, you then also get the habitat back for the animals and it provides them with food and shelter. So you say that the animals will then return to the regrowing area and help regenerate it. Finally, you can point out that the old growth area, so that's the bit in the middle, is never cut down and is a source of recolonising organisms. So there will always be plants and animals there that can move into areas that are regrowing. So each of these individual statements we've put down are worth a mark. You don't need every single one of them, we've got well more than 4 here, but if you have 4 individual and logical statements, you can get your 4 marks. Two of the substances the body excretes are urea and carbon dioxide. Complete the sentence, choosing the answer from the box. And this question is worth 1 mark. We are given the words carbohydrate, lipid, protein and salt. And the sentence we're given is, a person makes a lot of urea if the person's diet contains a lot of blank. so we just have to fill that in. And the answer is protein. Protein increases the person's urea production. Why must urea be excreted from the body? This question is worth 1 mark. The answer to this is that urea is a waste product. It means that it cannot be used by the body, and it is also toxic, so the mark scheme will allow you to describe this or state that the urea may damage cells or denature proteins. A person produces more carbon dioxide during exercise than when resting. Complete the sentences. Choose answers from the box. And this question is worth 2 marks. So looking at the words that we are given, we've got breathing, which is your inhalation and exhalation to exchange oxygen and carbon dioxide. We have osmosis, which is the movement of water across a partially permeable membrane according to a difference in water potential, which means that the water will go from a dilute solution into a less dilute solution. Then there is digestion, which is the breakdown of your large food molecules into small soluble molecules that you can absorb in your small intestine. Then there is respiration, which is your oxygen and glucose becoming carbon dioxide and water, and this gives you energy. And lastly, there is egestion, which is the excretion of waste substances. Now we can look at the sentences that we're given. So the first one says the process that makes carbon dioxide is blank, and we can fill that in with respiration, as when we look at that equation we see that CO2 is the product. The next sentence says during exercise extra carbon dioxide can be removed from the body by increasing the rate of blank. Now we're talking about the removal of carbon dioxide, and we can see that that happens through breathing because that's our gas exchange and we exhale carbon dioxide. Excess water must also be removed from the body. If a person sweats a lot, less water will be excreted in the urine. A healthy person did the same amount of exercise on each of 3 days. Table 2 shows the information for the 3 days. So here we have table 2, we have the days in the first column, air temperature in the second, the volume of water consumed in the third, and in the fourth there is the relative amount of urine produced by the kidneys. The question asks you to complete table 2 choosing answers from the box, and it's worth 2 marks. The words we are given are least, medium and most, and we have to insert them into the table. You can start with day 3, as that's the one that has data that is entirely different from the other 2, as day 1 and day 2 have the same volume of water. Day 3 has more water at 2,000 centimetres cubed, and it also has a lower air temperature. When the air is cooler, the person is less likely to sweat, and given that they are also consuming more water on this day, we can conclude that the relative amount of urine will be the most. With the next 2 options, the volume of water consumed is the same, so we need to work with our temperature values. On day 1, there is a higher temperature, and this indicates that they will probably be sweating more, and therefore have less extra water in their bodies, so this one will be the least amount of urine produced by the kidneys. And that means that the last one will be medium by process of elimination. Some people have kidney disease. Kidney disease may be treated by dialysis or by having a kidney transplant operation. During dialysis, a person is connected to a machine that filters the blood. Each dialysis session lasts about 6 hours, and the person has several dialysis sessions each week. Figure 12 shows how dialysis works. So here we have figure 12. We have a partially permeable membrane, and on the left is blood, and on the right is dialysis fluid. We also have a key which tells us what all of the little circles are representing. How does urea move out of the blood during dialysis? Tick 1 box, and this question is worth 1 mark. So the first option in multiple choice is diffusion. And if we review what that actually means, it's the movement of substances from an area of higher concentration to an area of lower concentration along a concentration gradient. and actually that is what's happening when we have dialysis. We have a partially permeable membrane which separates the blood and the dialysis fluid, and the blood is a substance that has a higher concentration of urea, and the dialysis fluid has a lower concentration of urea, so the urea will move across from the blood to the dialysis fluid along that concentration gradient. So the first option, diffusion, is the correct one. And to double check this, we can just look at what the other options mean. So digestion is talking about food, which is incorrect. Osmosis is when we're talking about water moving across the partially permeable membrane, so that's not it. And respiration is talking about your breathing and gas exchange, so that's not the answer either. Which substance in figure 12 does not pass from the blood into the dialysis fluid? Give the reason for your answer, and this question is worth 2 marks. So we work this out by looking at figure 12. We see on the diagram that the partially permeable membrane has gaps in it that represent the pores. We also see that on the left-hand side in the blood are large white circles that are larger than the pores. These large white circles represent protein molecules, which are large in themselves, and these are what can't pass through into the dialysis fluid. So the answer is protein, and the reason is that the molecules are too large, or that the pores in the partially permeable membrane are too small. 2 people have kidney disease. Person A is treated by dialysis, and person B has a kidney transplant. Figure 13 shows changes in the urea concentration in the blood of each person over 2 weeks. So below is figure 13. On the y-axis is the concentration of urea in the blood, and we can see that there is a solid line for person A and a dotted line for person B, and the x-axis shows the time in days. The question asks, how many dialysis sessions did person A have each week, and is worth 1 mark. Okay, so we're told to look at it in a weekly perspective, which means we're concerned with each set of 7 days. When we look at the graph for person A, we can see that there are sharp rises and falls in their line that represents the concentration of urea in the blood. Each drop in the concentration of urea must mean that dialysis has occurred, because urea has moved by diffusion from the blood into the dialysis fluid. So what we need to do is count each of these drops. So there's one between 1 and 2 days, another between 3 and 4 days, and another between 5 and 6 days. So that's 3. We can then check this by looking at the second set of 7 days, because we've got that time span. And again, we can see that there is 1 drop between 8 and 9 days, 1 between 10 and 11, and the last between 12 and 13. So that's also 3, and we can confirm the answer is 3. What happens to the concentration of urea in the blood between dialysis sessions? And this question is worth 1 mark. So we have another copy of figure 13 above. We've marked the dialysis sessions with our downward arrows, and so we just need to say what happens between them. And we can see that it is going upwards, which means the concentration of urea in the blood is increasing between the dialysis sessions. Give 2 reasons why kidney transplant is a better method of treating kidney disease than dialysis, and this question is worth 2 marks. So again, we're using figure 13 and we can see it above, and we want to compare the line for person A, which is the solid line, and person A is the one that's having dialysis, with the line for person B, which is the dotted line, so that person's had the kidney transplant. And the first thing we can see is that the line for person B is overall much lower than that for person A, which you have to take an average for because it fluctuates so much. So the first thing that we can say is that there is a lower concentration of urea in the blood for person B. We can also say that person B with the kidney transplant has a more constant urea concentration over time. Alternatively, you could look at the more logistical side of this. So the person with the kidney transplant has to have less hospital visits, and they don't have to spend time on the dialysis machine. They also have less restriction on travel, and they don't have the repeated piercing of the skin, which happens with dialysis and can increase the chances of infection. The transplant is cheaper in the long term, and lastly, it means that they do not have to undergo any dietary restrictions. Figure 1 shows a food chain in a pond. So here we have figure 1, the first is the algae, then a daphnia, a hydra, and then a dragonfly nymph. Which term describes the daphnia in this food chain? Tick one box. In this chain, the algae is what's known as the producer because it's making its own food and using the energy of the sun. Then the first thing to eat the producer, which is the daphnia, and what we're being asked about, is called the primary consumer because it's the first consumer in this chain. So the next one along would be the secondary consumer and so on. Draw a pyramid of biomass for the food chain. Label each trophic level. So we've got to draw a kind of diagram here, and it's referring to biomass. So this is the biological material that is derived from living or recently living organisms. We also have this term trophic level. So a trophic level is a group of organisms within an ecosystem which occupy the same level on the food chain. So they're eating the same kind of thing and they're predated on by the same kind of thing. When you're drawing your pyramid of biomass, the number of tiers that you've got, so the number of different levels, is representative of the number of different organisms you've got in your chain. So in this instance, we've got the algae, the daphnia, the hydra, and the dragonfly nymph. And then the widths of your tiers you've also got to pay attention to. So the bottom tier is going to be the widest, and then your middle tiers will be in the middle, and your top tier will be the smallest. So your diagram should look something like the one on the right, and you've also got to label each trophic level. On your lowest level with the most biomass will be the algae, then the daphnia above that, the hydra above that, and the dragonfly nymph at the very top. Give one reason why the total biomass of the daphnia in the pond is different from the total biomass of the algae. So the reason for this is that not all of the biomass is absorbed by whatever's consuming, and the mark scheme will allow you to go into some more specific details for this. So some of the algae that the daphnia is eating may be non-digestible. There may just be parts that the daphnia cannot use in its own processes, and so that will be lost in the feces or in the urea. And then it can also be used up in respiration, so the daphnia needs to use some up as energy for itself. And then a last reason that you might go for is that not all of the algae is eaten, so the daphnia may not have consumed 100% of all that biomass that it could be consuming. Students investigated the size of the population of daphnia in the pond. This is the method used. Collect 1 decimetre cubed of pond water from near the edge of the pond, pour water through a fine net, count the number of daphnia caught in the net, and then repeat steps 1 to 3 4 more times. Table 1 shows the results. So here we have table 1, and then on the left hand column there's the sample number, so 1 through 5, and on the right is the number of daphnia in 1 decimetre cubed of water. Calculate the mean number of daphnia in 1 metre cubed of pond water. 1 metre cubed equals 1000 decimetre cubed, and then we've got the table 1 on the left hand side to help us out. Now remember, to calculate the mean you've got to get the sum of the values and then you divide that by the number of values that you just added up. So here we're adding up the different numbers of daphnia in 1 decimetre cubed of water, so 5 plus 21 plus 0 plus 16 and plus 28. And we're including the 0 just because it reminds you that there are 5 readings and that should be included in your mean. So we divide that by 5 and the answer you get is 14. So this is our average number of daphnia in 1 decimetre cubed of water. However, looking at the line at the bottom, it says the mean number of daphnia in 1 metre cubed of pond water. So we've got to convert this value. and we've been given that 1 metre cubed is 1,000 decimetre cubed. So what we've got to do is multiply our 14 by 1,000. And the answer you get there is 14,000, so that's your final answer for this question. The pond was a rectangular shape, measuring of length 2.5 metres, width 1.5 metres, and depth 0.5 metres. Calculate the estimated number of Daphnia in the pond. Use your answer for the mean number of Daphnia in 1 metre cubed of water, and give your answer in standard form. So this question is worth 4 marks. For this, we're going to need the answer from the previous question. So the mean number of Daphnia in 1 metre cubed of pond water was 14,000, so we've got a note of that on the left. Now, volume is calculated by doing length multiplied by width multiplied by depth. So that's what we've got to do with the values the question gives us. 2.5 times 1.5 times 0.5 gives an answer of 1.875 metres cubed. So we know how many metres cubed we've got, and we know how many Daphnia are in 1 metre cubed. So the last step for this is to multiply the two together. So we do 1.875 multiplied by 14,000, and that gives you an answer of 26,250. So this is the answer as an ordinary number, however it says to give it in standard form, so we've now got to convert this. Standard form is written as y times 10 to the power of x, with y being a number between 1 and 10. To convert, we write down 26,250. We make a decimal point after the first digit there so that we make it between 1 and 10, so this will be 2.6250. And then we've got to count the number of digits after that decimal place to see how many orders of 10 we're multiplying by. So in this instance, we've got 1, 2, 3, and 4 after the decimal point, making this 2.625 times 10 to the power of 4. So that's the way you've got to write your answer. Now for this 4 marker, you get the first mark for calculating the volume of the pond, the second mark for knowing to multiply that by your 14,000 calculated in the previous question, another for getting that calculation correct, and the last for your standard form. Rainfall can cause fertiliser to be washed from farmland into a pond. The students investigated the effect of fertiliser on the population of Daphnia in water from the pond. The students put 20 Daphnia in each of 5 different concentrations of the fertiliser. The students counted the total number of Daphnia in each concentration of fertiliser after 2 weeks, and figure 2 shows the results. So here we have figure 2, and the y-axis shows the total number of Daphnia after 2 weeks, and then the x-axis is the concentration of fertiliser in milligrams per decimetre cubed. A concentration of 5.0 milligrams per decimetre cubed of fertiliser caused a large increase in the population of Daphnia. Explain why. So what's happened here is the fertiliser has fertilised the algae and it's caused it to start growing really quickly, and we've got lots of it in the pond. So this means that there's a surplus of food for the Daphnia and they're able to eat plentifully. Figure 1 is repeated below. So here we have figure 1, and we've got the food chain from earlier, the algae, the Daphnia, the hydra and the dragonfly nymph. And it says the population of hydra will decrease when 20 milligrams per decimetre cubed of fertiliser is added to the pond. Explain why. So if we have a look back at figure 2, we can see that where there is 20 milligrams per decimetre cubed in the last bar, the total number of Daphnia after 2 weeks is very low, it's the lowest on the chart. Then we can have a look at our food chain. So the algae is what's feeding the Daphnia and maintaining that population. The Daphnia is then feeding the hydra, and the hydra feeds the dragonfly nymph. And we know that at 20 milligrams per decimetre cubed of fertiliser, the population of Daphnia is very low, and this means that there's less food for the hydra, and so that explains why the population is going to decrease. Genetic material is made of DNA. Which structures in the nucleus of a human cell contain DNA? And the answer to this is chromosomes. Figure 3 shows parts of one strand of a DNA molecule. So here we have figure 3. It's given us empty labels X, Z and Y, and then we've got this phosphate group, which is the little white circle. Label parts X, Y and Z on figure 3. Choose answers from the box. If we have a look at figure 3, the top label is label X, and this is pointing to the sugar. And a way that you can identify this is by noticing that the shape is a pentagon, and that tells us that it's a pentose sugar, meaning it has 5 carbons. And in this instance, the pentose sugar is deoxyribose. The next label down is label Z, and this is pointing out a base. In DNA, the bases are adenine, guanine, thymine and cytosine, and this is represented by the letters inside each of the boxes. The bottom label, label Y, is showing you a nucleotide, and this is the combination of the phosphate, the sugar and the base. So these are the monomers that make up your DNA structure. A complete DNA molecule is made of 2 strands twisted around each other. What scientific term describes this structure? And the answer here is a double helix. DNA codes for the production of proteins. A protein molecule is a long chain of amino acids. How many amino acids could be coded for by the piece of DNA shown in figure 3? Tick one box. So an amino acid is coded for by 3 bases. So what we need to do is figure out how many sets of 3 bases are given to us in figure 3. We can see that there's 9 altogether, and that gives us 3 sets of 3. So that's the answer that you need to tick. Scientists have now studied the whole human genome. Give 2 benefits of understanding the human genome. One benefit you might mention is the diagnosis of genetic disorders. Another is the treatment of disorders such as this, so these could be inherited disorders or genetic disorders. And you only need 2 reasons to get these marks, but just as some alternatives, you could give understanding the evolution and ancestry, or the ethnic origins of humans. And lastly, you could use it to trace human migration patterns over history. Phototropism is a growth response by part of a plant to light. Name one other tropism. Give the stimulus the plant responds to in the tropism you have named. The first answer you might go for here is geotropism, and this is stimulated by gravity. Another tropism that the mark scheme will allow is hydrotropism, and that term hydro tells you that the stimulus is water. Another that you could say is thermotropism, and again the prefix thermo tells you that it's to do with temperature, and in this case it's heat. Plan an investigation to show the effect of light from one direction on the growth of plant seedlings. Include details of any controls needed. You may use some of the equipment shown in figure 4 and any other laboratory apparatus. And this question is worth 6 marks. So here we have figure 4, we have a lamp, several pots of seedlings, some scissors, a ruler, and we have cardboard boxes with lids. Now remember that this is a 6-mark question, so just listing 6 relevant points is not necessarily enough to get you all of the marks. Your method must lead to a valid outcome, and it must be sequenced in a logical order, because you're effectively giving the instructions for this experiment. A way that you can start off with this is by describing the setup of your experiment and also naming the control variables, the things that you're going to keep the same so that there's nothing else affecting your experiment. So you can say to use several pots of seedlings that will be given the same amount of water and also be in the same temperature and soil type. So here the relevant points you're making are that you're naming control variables like the same amount of water, and you're mentioning the equipment you're using, i.e. the several pots of seedlings. Now you want to set up your control part of the experiment. So we've got the same temperature and the same soil type. Now we want to have a pot that's got an even amount of sunlight on all area around it. So this is there for comparison against the other pots. And what we do with these other pots is we put them in our cardboard boxes. So these boxes will have their lids on the top, and then they will have a hole cut out on a specific side. And what this does is control the angle from which the light can shine onto the plant. So now we're beginning to describe our dependent variable, and this is the thing that we're going to measure throughout the experiment. So what we've got to do to be able to compare is measure the seedling height at the very beginning of the experiment before the light has been hitting them or they've had any time to grow in these conditions. So we calculate an average length for each pot by straightening the seedlings out against the ruler and then dividing by the number of seedlings we measured. Then you want to leave the seedlings for a sensible amount of time, so enough for them to grow a little bit, and you're going to measure again using the same method and calculating another average. The last stage of the experiment will involve calculating the difference between the heights of the seedlings before and the heights after, so this will be a mean increase. Now we're trying to measure the tropisms, and so what you can do is take a protractor and measure the angle of bending of the plant, because if you've got a cardboard box that has a hole in a specific side, the plant is likely to bend towards it, and you can measure how close that angle of bending is to the direction of light entry and see if there's a correlation. So this is how you get your mark for a valid outcome. Explain how phototropism in a plant shoot helps the plant to survive. This question is worth 3 marks. So in phototropism, the plant is inclining towards the stimulus, so the sunlight. So the plant leaves are receiving more light, which means that they can carry out photosynthesis at a greater rate. This allows it to then go on to make more glucose. And for that last mark, there are a couple of alternatives to glucose that the mark scheme will allow. Glucose is the monomer, however you can talk about the polymers, starch, carbohydrates and other organic materials.